All right. You guys have any questions over what we were going through last time? We had gotten to this point where we were going to start talking about DNA because you need to understand DNA and how it acts and gets replicated and everything else. To understand a couple of things, DNA is where the genetic coding that makes up genes is found. And so you need to understand something about the structure of DNA to understand what a gene is. But the genes, the sequences of nitrogenous bases within DNA basically specifies proteins. And proteins are ultimately what make you what you are. So they're one of the major building blocks of the bodies of organisms. And your ability to produce these proteins or not has a big effect on what you look like, how you act, et cetera, et cetera. And so, in order for you to understand certain things about DNA, though, we need to go back to a concept from basic chemistry that you may or may not have been exposed to in the past, but it has to do with what we refer to as hydrogen bonding. So you guys all know what water is, right? Two hydrogen atoms bonded to an oxygen atom. Well, when we draw atoms, we can use this thing called the Bohr model of the atom, which is not a very physically accurate way of thinking about an atom, but it's a good schematic way of thinking about an atom, because it allows you to think about the energy levels of these different electrons. So electrons have different levels of energy the further away they are from the nucleus of the atom. Oxygen has an atomic number of 8, meaning that it has 8 protons, 8 neutrons, and presumably 8 electrons. Hydrogen has an atomic number of 1, which normally means that it would have 1 neutron, but most hydrogen actually doesn't exist in that form. Most hydrogen exists as a single proton and a single electron, and there are isotopes of hydrogen that have a neutron in them, but most hydrogen exists like this, proton and electron. Remember that protons are positively charged, electrons are negatively charged. These electron shells get filled. The first shell gets filled with 2, the second shell gets filled with 8. And so if we put the first 2 electrons of the 8 electrons of oxygen into this first shell, it means that there are 6 in this next shell. But the way we fill these orbitals, these places where electrons kind of live in a Bohr model of the atom, is called orbitals. We put the first 4 in and fill each orbital halfway, and then we start filling in fully orbitals until we're up to 6. So now we have 1, 2, 3, 4, 5, 6, 7, 8. So we have 8 electrons here. But when your orbital is filled with 2 electrons, it can't bond with anybody else. And so in oxygen, we have, in this outer shell, we have 2 orbitals that are filled, and we have 2 orbitals that are unfilled. And it's these 2 orbitals that are unfilled that allow something like hydrogen to come here and bond with oxygen by sharing these electrons. So this is known as a covalent bond, and oftentimes when you hear people talk about covalent bonds, they're bonds where 2 atoms share their electrons with one another. That allows the outer shell of hydrogen to have a full orbital, and it allows all of the orbitals of oxygen to have full orbitals as well. So you should always expect oxygen, for the most part, to be making 2 bonds with other things. In the same way, when you look at carbon, carbon has an atomic number of 6, which means that it should have 1, 2, 3, 4, 5, 6 electrons. So now its outer orbitals are all incomplete. They don't have 2 electrons in them. And so in this case, you should expect carbon to make 4 bonds with other things. So where these electrons are located and how many of them you have determines what your bonding capabilities are with other things. And so you can just look at the structure of an atom and say, okay, I expect that atom to be able to make 4 bonds with other things. Nitrogen has an atomic number of 7, which means that you should expect 7 electrons. 1, 2, 3, 4, 5, 6, 7. This orbital is full, so how many bonds do you expect nitrogen to be able to make with other things? 3 generally, yeah. So a bond can be made here, here, and here. So knowing about electrons is super huge for thinking about the reactivity of atoms with one another. You know a little bit about their electron structure, you know a lot about their bonding capacity. The weird thing, though, is that that's not all you need to know. If you look at the periodic table, you'll remember that the periodic table looks something like this. Hydrogen is over here, oxygen is over here, nitrogen is here, and carbon is here. Helium is here, fluorine is here. Do you guys recall from chemistry, did you take chemistry in high school? Do you guys recall what electronegativity is? Do you recall hearing that term at all? Okay. Electronegativity is the tendency of an atom to pull electrons away from other atoms. So different atoms have different capacities. Some atoms are radioactive, some atoms have this characteristic of electronegativity. The higher your electronegativity is, the more of a tendency you have to pull electrons away from other atoms. And electronegativity increases as you go from the lower left to the upper right of the periodic table. And so one of the most electronegative atoms in the universe is fluorine. Fluorine and chlorine, right underneath it, tend to create ionic bonds. And ionic bonds are different because when you take sodium, that is full up in its outer shell. No, sorry. I got that wrong. Sodium has 1 electron in its outer shell. It's right below hydrogen over here. And chlorine has all but 1 of its orbitals full. Chlorine is so electronegative that rather than sharing its electrons with hydrogen's electron, what it does, then with sodium's ion, this is to make table salt. Table salt, sodium chloride is table salt. When table salt is formed, the tendency of chlorine to pull electrons away from other atoms is so great that rather than setting up a covalent bond where they share these electrons, chlorine strips the electron off of sodium altogether. Because sodium is now down a negative charge, it has a net positive charge. Now chlorine is negatively charged because it picked up an extra electron. It's negatively charged, and these 2 opposing charges are what holds sodium chloride together. This ionic bond is a bond that is formed when there's actually a transfer of an electron from one atom to another. Those 2 atoms become ions of different charges, and then those different charges are what attract the 2 atoms together. So when we make sodium chloride, we're making an ionic bond. So fluorine and chlorine tend to form ionic bonds, but the atoms right next to those tend to form covalent bonds, but there's an unequal sharing of these electrons. So a more productive way of thinking about an atom is at the core of the atom is where the protons and the neutrons are held. But then the electrons are, remember, these really tiny particles. They weigh hardly anything. And rather than thinking about them in these static orbitals, think of them as occupying space around the nucleus of the atom. They're basically zooming around randomly in the space around the nucleus. This is referred to as the electron cloud. And so this is the thing that blew my mind when I first learned about it. Here is a door. It is a solid door. You cannot run through the door. You can pound on the door. It actually hurts your hands. It's mostly empty space. It's mostly empty space that is occupied by these little tiny electrons. You have nucleus here, and you have all of this space in between them, and in that space are these little tiny particles zipping around in space, but because they're so tiny, they don't fill that space, really. They're just zooming around in their little trajectories. Impossible to know where exactly in this cloud of electrons a given electron is at a given moment. They're just zooming around randomly. What makes this door solid is the fact that these atoms are packed really close together, even though there's like this huge amount of space around each of them. But the other thing that makes them solid is that the atoms that are there are bonded solidly to one another. So what makes something solid isn't that you've packed those atoms in there to the point that there is no space in there. It's that they're packed in there in such a way that they're bonded tightly together that they basically become solid and immovable. What happens when you convert something from a solid to a liquid is you basically increase heat energy, which then drives those molecules further apart from one another, loosens them up a little bit, and then you go from a solid state to a liquid state. Heat them up even further, and you convert that from a liquid to gas where the molecules are spread very far apart. So what's making things solid isn't the actual physical space of the nucleus, it's actually the bond strength between these atoms and molecules. So why is this important? Well, what happens when oxygen and hydrogen hook up in water is that the oxygen atom, because it's electronegative, it holds its electron cloud in very tightly. And when it bonds with hydrogen, hydrogen has a single electron floating around it, but when it bonds with oxygen, it pulls that electron towards oxygen so that the electron is hanging out more in terms of its association with oxygen than it is with the positive ion, the positive proton that is in the hydrogen atom. Same thing with this hydrogen, with its one proton. It has an electron cloud, but that electron cloud gets pulled away from the nucleus of hydrogen such that there's way more negative charge over where the oxygen is than there is where the hydrogen is. So this thing becomes partially negatively charged, this becomes partially positive charged, and this becomes partially positive charged. And these partial charges, it's not a full charge like it's stripped away the electron like chlorine does, but it's a partial charge in that it's pulling those electrons of hydrogen further away from the hydrogen nucleus, making the oxygen a little more negative and making each of those hydrogens a little more positive. Now, why am I telling you all of this, is what you're asking. Well, when we have water molecules, what happens with water molecules is because each of these water molecules has a partially negative, oxygen bonded to two partially positive hydrogens, there are bonds that set up between these different molecules of water where the positively charged, slightly positively charged hydrogens bond with these slightly negatively charged oxygens, such that water takes on a structure such that the water molecules are oriented towards one another in particular ways because these oxygens always hook up with these hydrogens through what we refer to as hydrogen bonding. So these are weak intermolecular forces. They're weaker than a covalent bond. They're much weaker than an ionic bond, but they still bond together with one another in these weak ways that kind of provide structure, in this case, structure to water, molecular water. So hydrogen bonding is super important for you to understand like so much of biology at the molecular level. The reason that I am telling you this is that the structure of DNA is arranged such that you have two strands that lie next to one another. The components of those strands are held together with covalent bonds. You have a phosphate molecule, which is just a phosphorus with 4 oxygens around it. You have a sugar molecule here. In this case, the sugar is deoxyribose, which is why it's deoxyribonucleic acid. And then you have a nitrogenous base, and there are 4 different kinds of nitrogenous bases that we'll talk about here in a few minutes. But this phosphate bonded to this sugar, bonded to that nitrogenous base, and that sugar bonded to that phosphate, and that phosphate bonded to this sugar. Those are all covalent bonds. And those covalent bonds hold this strand of DNA together, and it holds this strand of DNA together. But the things that hold the two strands together, here in the middle, these are all hydrogen bonds. And you'll notice that these hydrogen bonds are due to, you have a nitrogen bonded to 2 hydrogens here, you have an oxygen here, you have an oxygen there, a nitrogen bonded to 2 hydrogens. Remember, nitrogen is right next to oxygen. It has a certain amount of electronegativity as well. And so these hydrogen bonds hold these 2 strands of DNA together to make the complete double strand of DNA. Because they're weak intermolecular forces, all it takes, if you're working in a lab, is a little bit of heat to take those 2 strands of DNA and split them apart. All it takes in a cell to split those apart is a little enzyme that facilitates breaking these 2 strands apart down the middle, kind of unzips them, and can zip them back up again because they want to be naturally zipped together anyway because of this hydrogen bonding that occurs. So in order to understand how you make new DNA from old DNA and how you make RNA from old DNA, you have to understand that it's easy to get access to those nitrogenous bases because the bonds that you have to break to get access to those nitrogenous bases are relatively weak. They're not super strong at holding those 2 strands together because all of those bonds are hydrogen bonds. Now, so before I go on, is everybody clear on that? What hydrogen bonds are and why they're important to understand how DNA is structured? Okay. So DNA is made up of a phosphate sugar backbone, and then bonded to each of these sugars is a nitrogenous base. So we have phosphate, sugar, and a nitrogenous base. And the nitrogenous base is called a nitrogenous base because it has nitrogen in it. And the nitrogenous bases are always bonded to the sugar part. And then these nitrogenous bases are bonded to other nitrogenous bases on the other strand of the DNA. And they can be bonded together either by 2 bonds, 2 hydrogen bonds. Let me draw them as dotted lines so that it indicates that it's not a covalent bond. Or they can be bonded together with 3 hydrogen bonds, holding these nitrogenous bases together. And whether they have 2 hydrogen bonds or 3 hydrogen bonds depends on the kind of nitrogenous base that's involved in this hydrogen bonding. So we have 4 different kinds of nitrogenous bases. There's another type of nitrogenous base, a 5th type, that is only found in RNA, which we'll talk about a little later. What do you notice about these nitrogenous bases? What makes these nitrogenous bases different from one another?
Some of them are guanine,
Okay, so they have different names, guanine, cytosine, adenine, and thymine, but what's different about their structures?
they're just flipped.
Some have
Like
more
the-
size than others.
Oh, go ahead.
Some have more size than others.
Okay, so this structure here, it has a 6-membered ring bonded to a 5-membered ring, okay? So some of them have a 5-membered ring attached to a 6-membered ring. Others of them are just a single 6-membered ring. What that means is that these are wider from side to side than these are. These are called purines. These are called pyrimidines. I always remember the names because the purine, which is the shorter name, goes with the wider, longer molecule. Pyrimidine is the longer name that goes with the shorter molecule. I never can keep them straight, so I finally found a way to keep them straight in my mind. All that matters to you, though, is knowing that purines and pyrimidines always bond to one another in this hydrogen bonding way. And so adenine is a purine. It always bonds to thymine, which is a pyrimidine. Guanine always bonds to cytosine. So in DNA, we have these, what we refer to as complementary bases. Complementary bases are bases that bond with one another. So they complement one another. Adenine always bonds to thymine, cytosine always bonds to guanine. The reason for this is that when you have an adenine, purine bonding to a pyrimidine, the space between the 2 edges of the molecule, between the 2 strands of the molecule, stays constant. And it doesn't matter whether you have a purine on one side and a pyrimidine on the other side, or a purine on this side and a pyrimidine on this side. The width of the molecule stays constant down the entire length of the molecule, because you always have a long one being bonded to a short one. Okay? But this complementary base pairing, adenine only has 2 places where it can create hydrogen bonds with thymine, which also only has 2 places where it can create hydrogen bonds. Cytosine and guanine have 3 places where hydrogen bonds can attach. So adenine and thymine bond with one another because they have the same number of bonds they're capable of making. Cytosine and guanine bond with one another because they have the same number of bonds that they can make with one another. So you're always going to have adenine connecting with thymine and cytosine connecting with guanine. And this way, DNA kind of structures itself. If you know half of a DNA molecule, you can infer what the other half of the DNA molecule is because of complementary base pairing. So why is all this important? definition of a gene, and just, I'm gonna tell you right now, I'm not gonna be happy with any of your definitions of a gene. So just prepare yourself for that. What is a gene? How do you remember talking about genes from your public school days? Just because I'm not gonna be happy doesn't mean you can't try.
Oh no, it's something passed down from your parents.
Okay, so one aspect of a gene is that it can be transmitted from one generation to another, sure.
Do you like that answer?
It's a super incomplete answer, but what you said is true of a gene. They can be transmitted from one generation to the next generation. That's just one aspect of a gene. Yeah.
Makeup of DNA.
Okay, so genes are made up of DNA. So they are DNA. What part of the DNA is important, though, for knowing what a particular gene is or does?
Genotype.
Okay, so genotype refers to, in diploid organisms, which we're diploid organisms, which means we have 2 copies of every gene. Genotype refers to, you have, let's see, how do I want to say this? Given that we haven't talked about genome structure yet. So let's say that you have, let's use a practical example. How many of you can roll your tongue? Let's see it. Oh my God, everybody in the class can roll their tongue. This is the first time you were rolling yours? Everybody in class can roll your tongue. So in tongue rolling, let's say that there is a gene for tongue rolling, because there is a gene for tongue rolling. And the cool thing about tongue rolling is the simple dominant recessive gene. And when you have a gene, we have the gene for tongue rolling. Genes have different forms. Somewhere out there, unfortunately for them, they're not in this awesome class of nothing but tongue rollers. There are people who sadly enough cannot roll their tongue. You ask them to roll their tongue and their tongue comes out and it looks like a cucumber or it's flat or something else. And try though they might, they cannot roll their tongue. And no amount of training will allow them to roll their tongue. They can't go to, like urban axes or something like that where you learn to throw axes. You can't go to urban tongue rollers and be trained on how to roll your tongue. You just don't have the right arrangement of muscles in the tongue to do that, because that arrangement of muscles in the tongue is determined by a gene. And in this case, the gene for tongue rolling, we could just designate it T. This is a dominant gene, which basically means that it's a trait that if the allele, an allele is just different forms of a gene. So in this case, we have 2 different forms of a gene. One form of the gene is the dominant form of the gene, which gives you the ability to roll your tongue. There's also a recessive form of the gene, which prevents you from having that ability. But you are always carrying around 2 copies of every gene. 1 copy of your genes you got from your mom, and 1 copy of your genes you got from your dad. And so you're always walking around with 2 copies of every gene. Now, for a dominant trait like tongue rolling, if you have 2 copies of the dominant allele, you can roll your tongue. If you have a copy of the dominant and a copy of the recessive, you can still roll your tongue, but because this recessive allele is recessive, you have to have both of them in order to be a non-roller. So I don't know what your genotypes are. So these are now genotypes. I don't know what your genotype is, but you can roll your tongue, so I know you're either homozygous, meaning you have 2 copies of the same gene, and dominant, meaning that those are dominant genes, or you are heterozygous, meaning you have 1 copy of one and then the other copy is the other. This is a homozygous also, but this is a homozygous with 2 copies of the recessive allele. So when we talk about genotype, we're talking about what's the complement that an individual has of these two versions of the gene. Do you have 2 copies of the same version? Do you have 2 copies of that version being dominant, 2 copies of that version being recessive? Or do you have 1 copy of one version and another copy of the other version, in which case you're heterozygous? So that doesn't really go into the definition of a gene. The genotype is when you have diploid organisms, organisms that carry around 2 copies of every gene, then what's the complement of those 2 copies? But the nice thing about not having the right answer is we have to cover a lot of vocabulary for you. You need to know what all of these things are for when we start talking about genetics. We're going to be referring to homozygous dominance and homozygous recessive and heterozygotes, and we're going to be referring to genes and alleles. So not useful for the definition of the gene, but certainly useful for getting us to cover some information. Who else wants to take a shot at the definition of a gene? So what is it about the DNA that makes a gene do what it does?
Characteristics of it.
What?
Determining characteristics of it.
It determines characteristics of what?
Offspring.
Okay, but how does it do that? So I mentioned that this allele gives you the ability to tongue roll. And I mentioned that this tongue rolling ability has to do with the structure of your tongue. So it's actually a physical capacity for rolling your tongue. How does the information in the gene that represents the dominant version of this gene, how is that version of the gene different from this version of the gene, which is what makes you a non-roller, which gives you a different structure of the tongue that results in non-rolling? What is it about the DNA? Is it the sugar, is it the phosphate, or is it the nitrogenous bases? Why would you say that? You just reasoned through it. You don't actually know that that's the answer. Why would the nitrogenous bases be the thing?
Because you could like rearrange them in different orders.
They're the part of the DNA that's variable. And so whether you have this sequence of nitrogenous bases on a DNA molecule versus this sequence. These are 2 different sequences. They would then code for different versions of whatever stretch of the gene exists. You'll notice that these things are largely congruent with one another, and they only differ from one another in this area in the middle. They're the same everywhere else. And this is what we find when we look at sequence level data for organisms, is a lot of organisms, especially closely related organisms, they're the same for large stretches of the genome, but there are particular places where the DNA sequence, the sequence of nitrogenous bases in DNA, varies from individual to individual. So a gene is a sequence of nitrogenous bases that vary from individual to individual potentially, but what do those sequences of nitrogenous bases do?
Provide information for your body to function in a specific way.
That's actually not bad.
Stupid science.
What's that? What's that?
My last name is Studebaker, and everybody always takes Baker and then switches up whatever. I said I was gonna change my name, my major from business to science. So
Because-
pseudoscience.
Oh,
Because it's
gotcha.
science.
So when I was in high school, I learned that a gene was a sequence of DNA, a sequence of nitrogenous bases in DNA that codes for a particular protein. Because when I was an undergraduate student and when I was in high school, it was all about proteins. We talked about protein. But the fact of the matter is that DNA can specify the order of amino acids in a protein, but it can also specify the order of nucleotides in things like RNA molecules, which are functional, but they're not proteins. And RNA molecules can have a huge effect on the phenotype, the final way an organism looks and functions, but they're not proteins. And so nowadays we say that it's a sequence of nitrogenous bases that codes for a particular functional product. That product can be protein, it can be nucleic acids, it could be, it could be lots of things. It could be what we refer to as transcription factors. These are things that turn genes on and off. And so they don't make proteins, but they tell genes to then be activated or inactivated to make proteins. And so they're signaling molecules. But they are also coded for by the sequence of nitrogenous bases in nucleic acids. So we have a more expansive view of what a gene is now than we did when I was an undergraduate student, but it's still the same in many ways. It's due to this sequence of nitrogenous bases that have some information content that the cell then interprets in making other products. Proteins, nucleic acids, transcription factors, things like this. So, adenine, cytosine, thymine, guanine is different than adenine, guanine, cytosine, guanine would be. That's a different information content. And you get all of this variability from gene to gene just by rearranging these 4 different nitrogenous bases. Okay? Questions about DNA? I realize I've had this slide up the whole class period, and that's all we've talked about is this one slide. Because these are fundamentals. The trick to understanding anything complicated is to get the fundamentals down. Once you have the fundamentals down, everything after that should be easier because you've engrossed yourself in the fundamentals. What was it, Yogi Berra? Who's the baseball player right here? Famous Yogi Berra quote, probably the most famous Yogi Berra quote. How do you win a World Series?
Practice makes perfect.
Nope.
Is it one game at a time?
No, although he could have said that. That sounds like a very Yogi Berra-ism. What's the secret to success in baseball? Pitching, catching, running. If you do those 3 things supremely well, you'll go to the World Series. Because it's basically batting, fielding, and then running bases. I mean, baseball, his point was baseball at its fundamentals is actually quite a simple sport. But mastering those 3 things is actually quite difficult. So mastering fundamentals is always going to make whatever you do next easier. I don't know, when you practice baseball, when you go to baseball practice, what do you do?
Throw, hit, and get ground balls, just that every day.
You don't just go out there and have some people playing defense and some people playing offense and play a game? No, you practice the fundamentals. Because if you get those fundamentals down, going out and playing a game becomes just second nature to you. All right. So DNA then comes in these long strands, super long strands. But we have to get those long strands shoved inside of a tiny cell. Not only do they have to be inside the cell, they have to be inside the nucleus, which is an even tinier compartment of the cell. So how do we do that? Don't worry about this as much. This is mainly to get you from the DNA molecule to the idea of a chromosome, so don't pay attention to the specifics of this. Know that this is where we're headed. What happens is this DNA molecule gets wrapped around little protein balls called nucleosomes, gets wrapped around those, and then those protein balls aggregate into little clusters, and then those little clusters get twisted around and folded up on one another until it gets to be a structure that's big enough that you can actually see it with a microscope that makes up this chromosome. So in this chromosome, you can take that chromosome and you can pull it apart, and the first thing that you're going to do is you're going to take these chains of nucleosomes, and these chains of histones, and you're going to unfold them, and then when you pull them apart even further, you're going to have little balls of histones with a little strand of DNA connecting them, and if you pull on it even harder, you would get the DNA to unravel from those histones would be released, and then you would just be left with this DNA molecule. And so you package DNA into a chromosome by winding it around little balls, squeezing those little balls together into a thicker strand, and then folding that thicker strand up on itself until it finally achieves the structure of a chromosome. And it's chromosomes that get transferred from one generation to the next generation that allows for this transmission of genes, because in those chromosomes are the DNA molecules, and the DNA molecules are where the genes actually reside. So there's this particulate matter, this particulate nature of inheritance where you physically take a bunch of genes all bound together on the same chromosome, and you move that from one generation to the next generation. But in order to do that, you have to make copies of the chromosome, which means you have to make copies of the DNA, because you can't take your cell's DNA and just off it to your offspring, because then you don't have any DNA. In the same way that when you're growing and you start off as a single cell, so this is one of the other most amazing things that you can ever learn, is that you start off with a single cell as a zygote. When your mom and dad had you, your mom produced an egg, your dad produced a sperm, your dad's sperm hooked up with your mom's egg, fertilization occurred, and at that point you become a zygote. The egg has half the genetic material of your mother, the sperm has half the genetic material of your father, so that when they come together in you, you now have a full complement of chromosomes. Your mom's egg had 23 chromosomes, your dad's sperm had 23 chromosomes, so that you could have 46 chromosomes when you were finally conceived. You become a zygote, and when you go through the process of beginning to grow into the human that you are now, the first thing that you do is you divide into 2 cells, then you divide into 4 cells, then you divide into 8, then into 16, then into 32, then into 64, then into 128 cells, and then 256, and then 512, and then 1024, etc., 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 until you get to what you are now, which is trillions and trillions of cells. We all started off as a single cell and then grew into these monstrosities that you see before you. Every time this happens, though, this cell and this cell all have 46 chromosomes. This cell, this cell, this cell, and this cell all have 46 chromosomes. So between the single cell stage and the 2 cell stage, you had to duplicate your chromosomes. But remember that your chromosomes are just DNA molecules and protein balls all bound up together in a chromosome, so you also had to duplicate DNA. Yeah?
When do we stop duplicating?
That's a complicated question. In some cases, you already have. So for example, nerve tissue is notoriously bad at making new nerve tissue. So once you have gotten your brain and your nerves and they're kind of in place, you don't turn over nervous tissue the way you do other types of tissues in your body. So when you do damage to nerves or do damage to your brain, oftentimes that damage is irreversible, because nerve cells have lost the ability to duplicate themselves. So the weird thing is, this zygotic cell, when you're a zygote, that cell doesn't look anything like a muscle cell or a nerve cell or an epithelial cell. It's just a generalized cell. It's what we refer to as a stem cell. Stem cells are these cells that have potential to become any cell in the body. And so when you are a zygote, that single cell has the potential to become any cell in the body because guess what? It produces daughter cells that eventually produce every cell in your body. And muscle cells are very different from nerve cells, which are very different from epithelial cells, which are very different from bone cells. What makes those cells different is which genes are being turned on in those cells and which ones are not being turned on in those cells. And so stem cells are important from a medical perspective because if you could go back to that early stem cell state, you could in theory grow a new kidney. If you had kidney failure and we found a stem cell that was capable of growing eventually into a kidney, we could just in a lab regrow a kidney. This is why people are interested in stem cell research, is because these cells have this potential to become anything in reality. So some cells don't keep that ability to continue dividing. Other cells do. You're constantly sloughing skin cells. That's why you should never commit a crime, especially a serious crime like rape or murder or something like that, because you are shedding skin cells constantly. And those skin cells, while most of them lack DNA, some of them have DNA, and they will find your DNA, they will put it into a database. And they will either find you if you have a criminal record because your DNA is already in the database, or if they suspect you, they'll collect a copy of your DNA, a sample of your DNA from a thrown away banana or the handle of your car door or whatever, and they will run that through the database and they'll find that your DNA matches somebody else's DNA and then you're screwed. So, don't commit serious crime because it's getting harder and harder to get away with it. Who was it? Oh, the Golden State Killer. You guys hear about the Golden State Killer recently apprehended in California? I'm the repository of all sorts of arcane knowledge. They found him because members of his family. So the Golden State Killer was this. He started out as the Bay Area rapist. He was a rapist who worked in the San Francisco area.
Was this guy who, his daughter, like, he was just on the news, I think, yesterday or something. I'm pretty sure I just watched it yesterday.
Yeah, tell us. What, I don't know if it's the guy. Tell me.
He was like a father to his daughter, he had like a family and everything, and he killed this like old lady, and he just like, it was like his, I forget his, it was like BTK or something, is that it?
BTK is different. That's here in the Midwest. This was a guy out in California. Anyway, before it was all over with, over a couple of decades, he killed people all up and down the west coast of the United States, mainly in California. And he started out raping people, and then somebody helped the police form a composite sketch, and he figured out that if he didn't start killing them, he would eventually be caught because people would be able to identify him. So he then graduated to murder after raping people. And there's just a book out. You guys know Patton Oswalt? His wife died, but she was an investigative reporter, and she was investigating the BTK killings, and she wrote a book about it that I just finished a couple of weeks ago. Anyway, they caught this guy because members of his family submitted their DNA to like 23andMe, these genealogy websites, and they had collected his DNA from crime scenes and from victims way back in the past, and they looked at the DNA in these genealogical repositories and found that there were relatives of the Golden State Killer who had submitted their DNA to these repositories. So not only, the whole purpose of those repositories is so that they can figure out who you're related to. And so even though he had never been convicted of a crime, he was actually, turns out he's actually a former police officer. Well, he had never had his DNA collected as part of a criminal investigation, but even though he didn't have his DNA collected as part of a criminal investigation, family members, extended family members of his submitted their DNA to these repositories. The FBI got access to those databases of DNA, figured out that there were some family members of the Golden State Killer in there. They then created genealogies, family trees of those people, and then identified people in those family trees that they thought might be good suspects, narrowed it down to 2 people. One of them they ruled out because he wasn't in the same place at the same time as many of the crimes, and so they narrowed in on the dude and found the dude. Yeah, question?
Oh, so when you submit your DNA to like 23andMe or those sort of things, it's like government access now?
No, but you sign away all of your rights, so you have to accept their terms of service, and if you read it, basically, you have to read terms of service carefully, because in those genealogical databases, if you click yes I accept, if you read down in there in the fine print, you're basically signing away your rights to your genetic information. And so you willingly gave up your genetic rights, your rights to your genetic information, so that when the FBI came calling, 23andMe was like, no big deal, they signed away rights to how we use these data. So, yeah, don't ever click accept without reading the fine print.
Did you read that? You actually read all of it.
Oh my God, when I went to buy my house, have you ever signed a mortgage?
Not yet.
Oh my God.
No.
It's like 150 pages of legalese. So I bought a house in 2004 here in the historic district. I live about nine blocks away, and I went to the mortgage office, and he like the mortgage officer scheduled like 15 minutes to get us in and out. I'm like, okay, let's see the mortgage, and he's like, okay, all you have to do is sign where the little post-it notes are. I'm like, nope, not gonna do it. And so I sat there at a table in his office, and for like 3 hours, read every page of the mortgage document, and when I got to a paragraph that I didn't like, I just drew an X through it. And I called the mortgage officer and said, I don't agree with this. And it was all kinds of stuff, like, part of it was giving away private information. I didn't want to give away my private information so that City Mortgage could do whatever they darn well pleased with. So I just crossed that out, called him in and said, I don't agree with this. And then he would read it, and he's like, well, it's a standard boilerplate mortgage. I'm like, yeah, that has a clause in it that I don't like. Either sign off on it, or I'll go get a mortgage someplace else. So I put my initial there, he put in his initial there, and continued going through it, over page after page after page. Like 3 hours later, he was willing to sign off on anything just to get me the hell out of his office. And I'll bet I got rid of probably 15, 20 paragraphs that had things in it that I didn't agree to in terms of the terms of the mortgage, how it was gonna be paid off, what they would do with my private information, et cetera, et cetera. Yeah, yes, it's a total pain in the butt to read those things, but in some cases it can cause you financial troubles down the line, because, I don't know, there was something in there about missed payments, which I haven't missed a payment ever since, but in the event that you do, they can do all kinds of things to you. They can ramp up the percentage rate of the mortgage because of missed payments. They can do this, that, and the other thing. And I'm like, no, I really don't want you to do that to me. No, don't want you to do that to me. And he came in and he initialed, and I initialed, and then that was gone from the mortgage. But yeah, my mortgage document is like that thick. I have it in a folder in my filing cabinet at home. And then they sold my mortgage, so Citicorp sold my mortgage to another company, because that's what mortgage companies do. They sell assets, and mortgages are assets. And I had to contact the new company and basically say, you now own my mortgage, you need to look at that mortgage carefully, because there are a lot of things in that mortgage contract that you probably are unhappy that Citicorp like X'd out. And I'm going to hold you to those same agreements that I had with Citicorp. Just buying my mortgage doesn't mean that I now agree to whatever your mortgage terms are. I agree to the mortgage terms that are in my mortgage document. So yeah, it's first day of class, what did we talk about? Or second day of class, what did we talk about? Oh, you've already forgotten. We talked about soybeans. Close. Talked about soybeans and tariffs, and I think it was you who commented, like, how long did it take you to go through that? I'm like, hours. It took me hours to put that together, because I put it together new every year. Living life this way is exhausting. Not for the faint at heart, but think about what you're giving up. So have you done 23andMe,
No.
may I ask?
No.
Oh, but now you're probably not, are you?
No, I'm not going to.
Okay.
That's not the right to kill.
Yeah. Yeah, so,
You're the killer.
so no, I think it's crucially important. So this is the critical thinking college. How many service agreements have you just clicked on without critically evaluating what it is that you're agreeing to? Yeah,
Everyone.
yeah, every one of them. So these guys have the potential. So you're sloughing off epithelial cells, you're sloughing off the cells inside your gut. When muscles get damaged, you can build new muscles as a result of that damage. Your bone is constantly reconfiguring itself. There are cells in your bone that eats up the old bone, and there are other cells that add new bone back, which is why you can heal from bone fractures throughout your life, is because you have those cells that have that capacity. But not all cells can do that. So you go through this, and as you make new cells, you have to create copies of your DNA so that you have enough DNA to distribute to the new cells. And so every bout of cell division involves copying DNA. So you have two copies, so you can send one copy to one cell and another copy to the other cell. And your cells just do this over and over again during the process of cell division. Other questions? All right, so we can actually look at your chromosomes. We can stain them with different colored fluorescent dyes. We have 23 chromosomes, 22 autosomal chromosomes, and 1 pair of sex chromosomes that are involved in determining whether you're male or female. These occur in pairs. And so when you look at these things, you can also stain them with a different kind of dye that can give the chromosomes banding patterns. And so these chromosomes occur in pairs, and these pairs of chromosomes are called homologous chromosomes. Once again, this is a term that you need to know in order to work through genetics. So homologous chromosomes are just chromosomes that have a particular structure. So they're long. In some cases, the centromere is located near the ends. In some cases, the centromere is located more in the middle. But you have, in this case, there are 23 pairs. They're not illustrated as pairs here, but there are 23 different types, and then 2 different types of sex chromosomes. So for that chromosome 1, you have one of these that you got from your male parent, and you possess one of these that you got from your female parent. And homologous chromosomes, they're not just the same in length and where the centromere is located. They're also the same in that if the gene for tongue rolling occurs here on the chromosome, the gene for tongue rolling occurs in the same place on the other chromosome. So there are chromosomes that not only are similar in structure, but they also have the same genes. Now, they may have 2 different versions of that gene. Remember, this is the dominant allele, and this is the recessive allele. But these are dominant and recessive alleles for the gene that determines tongue rolling. Okay? So the tongue rolling gene will be there. The genes for eye color may be somewhere else. Eye color is determined by multiple genes, so eye color might be determined by a set of genes someplace else. But if the genes for eye color are here on this chromosome, they'll also be at the same place on the other homologous chromosome. So the homologous chromosomes that you possess came from your dad's sperm and your mom's egg, came together in you to make you who you are. If you possess this combination of alleles, then you are a tongue roller. If you got a recessive allele from each of your parents, then you're not a tongue roller. This is why you don't look exactly like your parents, because you get half of their genetic information, you get half of the other genetic information, and how those 2 forms of genetic information come together in you determines what you look like. So you might resemble your parents in some ways, but you're also going to be different than your parents in some ways, in the same way that you resemble but are different from your siblings. Okay? So it doesn't matter whether we illustrate these things this way or whether we illustrate them after they have been duplicated. So when you go through this process of creating new DNA, you also create new chromosomes, such that this chromosome will now have a second arm, but that second arm will not only have the same genes in the same order, but the alleles will also be the same, because this is just a copy of this. This copy was made from this copy. In the same way, if you had a dominant allele here, when you make this copy, it will also have the dominant allele here. So now, when you take one of these, take this one for example, and partition them into a new cell, the new cell has the same genotype as the old cell. When you take these two and partition them into a new cell, the new cell has the same genotype as the old cell when we go through cell division. So they go through this copying. These are the chromosomes before they've been copied. 2 homologs, 2 homologs, 2 homologs, 2 homologs, 2 homologs. These are the chromosomes after they've gone through the process of copying. 2 homologs, 2 homologs, 2 homologs, 2 homologs. But these 2 homologs don't possess 2 copies of DNA. These 2 homologs now possess 1, 2, 3, 4 copies of DNA. So now you have 4 copies of DNA, so that when that cell divides, you put 2 copies into one cell and 2 copies into the other cell. And with that, we're out of time. Yeah, question?
In stem cells, is every type of like information that could possibly become you present in that like DNA strand?
Yeah, so the stem cell has your complete genome in it. It's just that in this genome, you have all the different genes with whatever variation from one chromosome to another chromosome of the homologs is there. And so what makes that stem cell, a generalized stem cell, turn into a muscle cell is that not all these genes get activated. So if you're creating a muscle cell, the genes for eye rolling, or sorry, not eye rolling, that's what I do a lot of. The genes for eye color don't ever get turned on. And so you don't produce pigment in those muscle cells. You produce other things like myosin and actin and things like
But
that.
they're still there.
Those genes are all still there. It's just that in one cell type, some genes get activated and others don't get activated. And in other cell types, a different set of genes gets activated. Yeah.
When does like someone stop having stem cells?
That's the tricky thing about stem cells, is you lose your stem cells very early on in development in many cases.
Can you like extract them while you're like?
So in theory, you should be able to extract them and freeze them. I don't know of a lot of people who have done that. There are some people who will freeze their umbilical cords, because that possesses fetal stem cells from the baby when you give birth and things like that. The trick though is if you haven't done something like that, and also the other thing is those things might or might not be viable once they're thought out.
Yeah.
Human cells aren't like worm cells and bacterial cells. You can freeze them for long periods of time and then just warm them back up and they'll start dividing. So the trick is, how do you get normal cells to become stem cells once they've lost that ability? These stem cells are called pluripotent. Because they have the potential to turn into lots of different things. And so the trick is, how do you get a mature cell to regain that ability? And that's actually an area of active research in stem cell.
Do you need these?
Yeah, I do need your